to talk today. What I thought I would do is talk with you a little bit about what we're learning about the genetic basis of bipolar disorder. What are all these things that we keep hearing about the effect of genes on bipolar disorder and other psychiatric disorders? What does it all mean? So let's get started by just looking at a few questions that I hope to address today. And I'm seeing if my slide will advance here. There we go. Okay. So the first is, do genes influence bipolar disorder? What evidence do we have for that? And secondly, if we think that's the case, are we able to actually identify genes that may be contributing to the disorder? And then can this kind of research tell us something about the boundaries of psychiatric disorders? That is, how we think about the distinction between, say, something like bipolar disorder and major depressive disorder, or schizoaffective disorder, and so on. So I'm going to approach those questions and look forward to taking questions from you as we get towards the end of this. The first thing is the question of what role do risk factors play in the development of bipolar disorder and other psychiatric disorders? If we asked what are the most influential causes of these conditions, what would people say? Well, if you asked psychiatrists 50 years ago that question, the answer would often have something to do with parenting, and specifically mothers. And so we would get views like the ones that you see on this slide, which were not fringe ideas. They were in many ways mainstream ideas, that things like autism or schizophrenia, for example, were caused by some lack of enough affection on the part of a mother. And people can imagine what kind of a burden and pain this view might entail for families and people suffering. And it really was the development of a better understanding of biological and environmental and genetic risk factors that began to overturn some of these ideas. And in fact, as people probably know, in the last few years, we have just seen a tremendous acceleration of our understanding of the genome, the human genome. And it has been a tremendous decade, beginning with the sequence of the human genome about a decade ago, and then several big science projects that have told us a little bit about the kind of genetic variation that we see across the genome around the world. And those studies, one called the HapMap project and another one called the 1,000 Genomes project, have led to the development of tools that allow us to actually study the genome in a very detailed way. And then more recently, there has been a focus on understanding what the genome is really doing. What are the functional elements, in addition to the genes that we've kind of known about before? What is all this DNA sequence doing? And that's a very active area of research. Well, there has been a lot of interest in identifying potential genetic contributions to psychiatric disorders. And when we think about the question of, do genes play a role in a condition, whether it's bipolar disorder or asthma or anything else, we typically ask a series of questions that involve a series of study types to answer those questions. And the first question is often, does this condition run in families? The second is, if we find that there is evidence that it's running in families, it's more common in family members, let's say, than in the general population, then we might ask, well, how much do genes actually contribute to that similarity in families? And then we might go on to ask, well, if there is a genetic contribution, what genes are actually involved? Can we map them and identify them? And then finally, if we do actually succeed in that, how are these genes actually translating into risk for disease? And so those are the questions that you see on the left side. On the right side are the study designs that people typically use to answer those questions. So let's just review that briefly for a second, especially with respect to bipolar disorder. So does it run in families? And here, the way researchers tend to answer this question is to do a family study where you're essentially comparing the risk or frequency of the condition in, say, first-degree relatives, that would be children or siblings or parents, of somebody who has the condition. We call that person an affected proband, to the the prevalence or risk for relatives of people who don't have the condition. So how much more likely are you to have it if you have a first-degree relative with the condition? And sometimes we talk about the recurrence risk ratio, which is similar to this. It's basically the risk in a sibling or other first-degree relative of the condition compared to the risk if we chose you at random from the population. How much more likely are you to have it if somebody in your family who's close to you and your immediate family has it? And what I've shown you on this slide is just an example of the answer to that question for a number of conditions. And they range from very high numbers. So anything that's significantly higher than 1 means that the ratio is greater than 1, that there's some evidence that it's running in families. And what you can see is that for something like autism, let's say, that might be somewhere on the order of 50 to maybe even 100. For bipolar disorder, for schizophrenia, for eating disorders, actually, it's about a 10-fold increased risk. And for things that are maybe a little bit more common, like depression and anxiety disorders, the increased risk is not quite as high on a relative basis, but it is still running in families. It's still greater than 1. And in fact, all of the neuropsychiatric disorders that have been studied do appear to run in families to a degree. The next question is, things could run in families for reasons that don't really have to do with genetic similarity among family members. So how do you really tease that apart? The fact that it runs in families could, in principle, be related to something about the family or the environment, and not really about genes. So one way that people have typically answered the question of what's the contribution of genetic influences is to do something like a twin study. And here, as people know, there are two kinds of twins. There are what we call monozygotic, that is identical twins, and there are dizygotic or non-identical twins who are basically about as similar genetically as any other siblings. So if you now ask how often, if you have one twin who has the condition, let's say it's bipolar disorder, how often does their twin also have the condition? How concordant are they for it? And if they are genetically identical and their concordance rate is higher than genetically non-identical twins, you can infer that their greater similarity with respect to the illness has to do with their greater genetic similarity. And so that implies that there is a genetic contribution. You have to make certain assumptions. You have to assume that, for example, identical twins are not treated by the environment in some way more similarly than non-identical twin pairs are, because that could make them appear to be more similar for reasons that are not related to their genetics. And if the identical twins are not 100% concordant, that is, not every time one of them has, say, bipolar disorder, the other one does, that implies that it's not all about their genetics because they are identical twins, essentially. And here we sometimes talk about a number called the heritability, which is a complicated, a deceptively complicated notion. And it's really technically the proportion of variation or variance in the population. So think of that as the proportion of risk of the illness across the population that's attributable to genetic variation, to genetic differences between people. And that number could range from 0, that is, none of the differences in who becomes ill, none of that risk is due to genetic differences between people, or it could be 100%. That is, it's all due to genetic differences. And the answer, of course, is that it's neither. It's never either 0 or 100%. But once again, all of the psychiatric disorders that have been studied appear to have a heritability that is at least moderate. So what you see on this slide is heritability estimates for a variety of neuropsychiatric phenotypes or conditions. And you can see that for things like autism, schizophrenia, bipolar disorder, that number is pretty high. Similarly for ADHD. More moderately for things like alcohol or substance dependence, eating disorders, and maybe more modestly for depression and anxiety disorders. And at the bottom, I I included the number for breast cancer because it illustrates a point. We think of breast cancer as certainly having a genetic component, and in fact, specific genes or mutations that can cause breast cancer were identified quite some time ago. And yet its overall heritability is lower than the psychiatric disorders that I list here. And that tells you something about heritability, because heritability doesn't really tell you much about how influential any given genetic risk factor is. It's just sort of the overall contribution of genetic differences across the population. So even though there might be some mutations that have a big effect on breast cancer, if they are not a very common cause across the population of breast cancer, they may not contribute that much to the overall heritability. But the bottom line here, I think, is that we now have seen fairly consistently in twin studies that the heritability of bipolar disorder and mood disorders and psychotic disorders and a whole variety of psychiatric disorders can be moderate to quite substantial, which tells us that there are genetic variations that one should be able to identify that contribute to this overall estimate of heritability. And so that brings us to the question of, can we actually identify and map some of these genes? And just to bring you back to high school biology or wherever you may have seen this kind of thing, as you know, our genomes are really a collection of chromosomes. We have 23 pairs of chromosomes in most of the cells of our body, with a few exceptions. And the chromosomes are really long strands of DNA that are also wound up with some proteins. And the DNA is a long molecule that has a sequence of letters or bases, A, T, C, G. And those letters are sometimes organized into functional units that we refer to as genes. And genes typically make proteins that run the machinery of the cell. Sometimes they make RNA, another type of nucleic acid like DNA. And it turns out that there's a lot of other stuff outside of the genes themselves that is very active about our genome. A lot of it has to do with regulating when genes turn on and turn off and so on. But really, when we're doing these genetic mapping studies or molecular genetic studies, we're trying to ask, are there regions of the genome or DNA variations that seem to actually confer risk to whatever the condition that we're talking about is? And one of the key points to keep in mind as we think about these kinds of studies is that psychiatric disorders are what we call complex. It's not the kind of situation that we may be used to thinking of when we think of genetics, something like a dominant disorder or a recessive disorder where you inherit a mutation, let's say, from a parent or two parents, and then you develop the disease. And I've given the example on the left side here of Huntington's disease, where there might be a mutation in one parent that is that sort of orange oval, and the offspring in this pedigree or family who inherit that develop the disease, which is indicated by the dark shading. And in this case, because it's dominant, you just need one copy of this mutation and to develop the disease. The recessive would mean that you'd need one from each of your parents. But we don't think that's what's happening. In fact, we know that's not what's happening in common situations of psychiatric disorders. It is not due to a single gene. There may be hundreds, maybe even thousands of genes that may contribute to some degree, each of which may have a small effect on its own, and each of which may need to interact with other genetic variations or with environmental or experiential exposures in order to actually increase the risk of a disorder. So that's really important to keep in mind, because we're not talking about identifying the gene for something or the mutation for something. It's a much more complicated picture than that. Now, when we talk about actually identifying some of the variants that may be involved, there are a number of things we might mean by that. There are different kinds of genetic variation that we talk about, and this is an example of some of them. the most commonly studied kind has been single nucleotide variants. What that means is single letter changes in the DNA. So one person might have a C at this position in the genome, and the other person might have a T. So that's a single base change. There are also, as you see on this slide, other examples where there's a slightly longer stretch, or in some cases a much longer stretch, in the case of copy number variants of DNA that may be involved. And when we talk about copy number variants, we're talking about, in some cases, 1 million or more letters of DNA that may be either missing or duplicated. And it turns out that these are relatively common, actually. They're not necessarily deleterious, but they are a cause of a number of conditions, including psychiatric disorders in rare cases. So these are the kinds of things we're asking. Now, the other thing to keep in mind is that these studies, which for a long time we had to do kind of one by one, that is, to look at one variation at a time and ask the question, is it possible that this might be related? And we had to guess beforehand which one to look at, just because of the technology, has now really changed. And we are now able to do what people call genome-wide association studies. That is, we can ask whether there's a correlation between a genetic variant anywhere in the genome and whatever it is that we might be interested in, whether it's depression or diabetes or heart disease. And we can now survey the whole genome in one experiment. And so what we're asking is there, for example, a single base change that is statistically more common in people who have the condition than people who don't? And just to underline that a little bit and maybe demystify these studies, you know, the genetic research is sometimes seems quite complicated and hard to follow, and you know, there's a lot of technicalities to it. On the other hand, at its essence, many of the studies that people do boil down to the kind of studies that many of us are familiar with in other settings. So, for example, if you wanted to know, does smoking increase the risk of heart disease or heart attack? One way to do that is to identify a number of people who have developed a heart attack. And those are the people that we're calling cases here. And then a number of people who have never had a heart attack, which we'll call the controls. And basically what you do in this kind of study is ask, is smoking more common among the people who've had a heart attack than among the people who haven't? We call this a case control study. And you know, you can do this with smoking, with all kinds of other epidemiologic risk factors, and this is in fact how many of them have been identified. So that same basic principle is what we're doing in these genetic association studies, except substitute for the cigarette DNA differences. So now you're asking, are there alleles or variants that are more common among people who, let's say, had a heart attack than among the people who didn't? And you essentially do the same kind of analysis. There are a lot of nuances to it, but that's essentially what you're asking. So for a long time, there had been many studies which were really hard to replicate. That is, the results would be reported, and people may remember this, that you know, there'd be a headline, the gene for bipolar disorder has been found, or the genetic variant's been found, and then you wouldn't hear that much about it, and they were hard to establish. In this new era, with much larger studies and the ability to look across the genome without having to place a bet about where to look beforehand, the situation has really changed quite a bit. And one of the first success stories in that way, in fact, was bipolar disorder. And this is a paper that was reported now about 4 or 5 years ago, in which researchers from the United States and the UK got together and did a genome-wide study of bipolar disorder and identified 2 loci, two genes essentially, that appeared to be strongly associated with risk for the disorder. That is, statistically strongly. That doesn't mean that each individually carries a huge effect, but the statistical evidence was really much stronger than what we had seen previously. And the names of these are not terribly important. One of them is known as ANC3, and the other one is a, a calcium channel subunit in the brain. And there are a couple of things to remark on here. One is that it required much larger study size to pick up these effects in this very statistically strong way. And that relates back to the fact that we think that each of these variations may carry a very small effect on their own. So you really need very large studies to pick up those effects. But the other thing is that they pointed us to a couple of genes which people hadn't really been thinking about before for bipolar disorder. Most of the guesses that people had made had to do with genes related to how drugs work, for example, how antidepressants work, or how lithium works, and so on. And these were sort of out of left field. That is, people, they hadn't been on our radar screen as potentially contributing to the condition. And that was very good news, because it meant we were learning something fundamentally new. And, excuse me, the value of that is that it might point us in totally new directions in understanding the biology of bipolar disorder. So this study was one of several that really began to suggest that we were in a new era in terms of the genetic research in psychiatry. And remarkably, you could say that before 2008, virtually no specific genetic risk factors or genetic variants had been identified sorry about that, for common psychiatric disorders, including bipolar disorder. And that's, you know, after a couple of decades of work. Remarkably, in just the last few years, that situation has changed quite a bit. And you can see from this slide this remarkable uptick in the number of gene findings that really are pretty statistically strong and people feel quite confident about. And that number is only increasing, in large part, again, because of the size of the studies that are being done, both in terms of the number of people who have participated in these studies and also the technology and our ability to search the genome. So we now have a growing list of specific variants that have been associated with bipolar disorder, schizophrenia, autism, and so on. And this is actually, this table here is a little bit outdated. There's quite a long list now, and this is really just for illustration purposes of a couple things. The top part are regions of the genome. So if you see something like 15q11-13, what that means is that there's a region on chromosome 15 on what we call the long arm of that chromosome that has a copy number variation that's been associated with autism. And chromosome 22, Q11, there's another one. Now, what's interesting about this is if you look at the frequency, it's 0.3% of people with schizophrenia in these studies, so less than 1%. The odds ratio, that is the increase in risk, essentially the relative increase in risk, is about 20, so maybe about a 20-fold increase in risk. Whereas if the red variants at the bottom of the table, the bottom half, are SNPs, single nucleotide polymorphisms, that go by funny names like RS109943336, etc., in and around genes like ANC3. But if you look at those red numbers, the frequency is higher. These are common. These are things that people commonly carry. But they also have a much, much lower effect individually. So maybe something on the order of 1.2 instead of 14 or 15. And you know, these numbers are not definitive numbers. These come from some studies, but they give you a flavor of the fact that, you know, some of these common single nucleotide polymorphism genetic variants, which are the ones that we often hear about, by themselves really only contribute a small amount. What they do is point us to new directions in biology to understand how is a gene like this involved in risk for the disorder? What's the pathway involved? How does that relate to brain function or to psychological function and so on? Now, we're entering another advancing age of genomic science, which people are excited about, but we're still at the early stage, and that is actual genome sequencing. Instead of sort of looking at even 1 million variations across the genome, it has become feasible to actually look at all of the variation and actually sequence each genome. And the cost of sequencing has dropped dramatically in the last 15 years or so. On the order of what you're looking at in this slide is Moore's law, that white diagonal line. That is, you know, it comes from computer science of the doubling of a computer chip's ability every 18 months, which we've seen happen over the last 15 years or so. But then look at the cost of sequencing a genome, and you see it's dropped even much faster than that. And so it has now become feasible for several $1,000s or perhaps even less, depending on how extensively we do the sequencing, to really look at all of the variation. And we think that is going to begin a whole new series of studies that should tell us something even more detailed. So we're still in many ways at the very start of this, but the fact that there have been some successes already in the last 5 years and that those are accelerating is really, I think, cause for optimism. The other question I wanted to pose, because I think it's both interesting and important, is whether this kind of research tells us something about what I've referred to here as the phenotypic boundaries. That is really about how we diagnose and classify psychiatric disorders. And what I've said here is, does DNA respect DSM? That is, do the genetic risk factors actually map onto the diagnostic categories that we have? And as you undoubtedly know, the primary system for classifying or diagnosing disorders that psychiatrists, at least in the United States, have used has been the DSM system. And what you're looking at in this slide is for the DSM-1, the one all the way on the left at the bottom, DSM-2, 3, 3R, and 4, and you know that next week or the week after, the DSM-5 is going to be released. What you're looking at here is the number of diagnostic labels in that blue line and the number of pages in the book. So there has been a tremendous growth in the number of diagnostic categories, and it does shift. And in fact, if you look at the history of the evolution of how we've diagnosed or thought about psychotic and mood disorders, much of it is traced back to Emil Kraepelin, very influential psychiatrist more than 100 years ago, who distinguished manic depressive insanity, as he called it, from dementia praecox, which was later called schizophrenia by Eugen Bleuler. And then it wasn't really until the late 50s or 60s that bipolar disorder and unipolar or major depressive disorder began to be distinguished. And then there was the distinction between bipolar 1 and 2 disorder. Schizoaffective disorder became part of the DSM system, and bipolar 2 and DSM-4 as well. So there's been this kind of evolving picture of how we even think about classifying these disorders. So one question is that how the genetic and family risk factors really pan out? Do they fall out into these sort of distinct categories? And there's a lot of research in this area, and I'm not going to try to even summarize all of it. I'm showing you data from one study that was published a couple of years ago just because it was such a large study. This was of a Danish population-based cohort, basically a birth cohort of everybody born between 1980 and 94, nearly 900,000 people. And what you're looking at here is the risk of, or the increased rate, relative rate of a variety of disorders on the left: schizophrenia, bipolar disorder, schizoaffective disorder, affective, anxiety, personality, substance abuse. And across the top is whether one has two parents with a serious mental disorder, which they classified as affective or non-affective psychosis, or one parent who has that and another parent has another psychiatric disorder, or one non-affective psychosis, or et cetera, all the way across the top. And so compared to having parents with no psychiatric disorder, you do see an increased risk of a variety of conditions. And what's interesting is that the risk of a whole range of conditions is elevated. It's not as though specific diagnoses are tracking in families, but there is in fact an increased risk for a number of conditions. And that fits well with what we've seen in twin studies as well. And in fact, in twin studies, you can get an estimate of what people call the genetic correlation. So that's asking how much of the genetic differences that confer risk to one disorder are correlated with, or you can think of as kind of overlap with, the genetic risk factors that contribute to another disorder. And so what you see on this slide are these numbers. So that the correlation could be anywhere from 0, that they're completely uncorrelated, to 1, which would mean essentially 100%. And what you see here is that, and these are data from twin studies, that these conditions are genetically related, at least on the basis of these twin studies. So bipolar disorder has genetic overlap with schizophrenia, depression. Depression itself seems to be related to a variety of anxiety disorders and ADHD, and you can see the rest here. And in fact, if you tried to draw the DSM based on what the family and twin studies seem to suggest, you wouldn't so much see these distinct categories as a series of overlapping categories to some degree. There is certainly differences, but the boundaries are not all that sharp. And that's both interesting and important for a number of reasons, and it also has motivated people to begin to look at whether some of the genetic risk factors that have been identified are actually conferring risk across different diagnostic categories. And one of the first hints that that was the case came from studies of something I referred to before, copy number variation. And these copy number variations are deletions and duplications of up to, say, 1 million letters of a DNA sequence. They may be slightly more common, for example, in the offspring of older fathers. And they seem to, on their own, have a larger effect on risk for disorders than some of these SNPs that we talked about. But they tend to be quite rare relative to these SNPs. So typically the ones that have been linked to, say, schizophrenia or autism might only be seen in 5% of cases if you put them together. But one of the most fascinating findings that's emerged as people have studied these copy number variants is that they do seem to have broad effects. That is, they have been linked to a whole variety of different neurodevelopmental conditions. So what you're looking at here are the copy number variations in the column on the left. So chromosome 1, long arm 21.1 seems to has been identified as a risk factor for autism, for intellectual disability, schizophrenia. There's, for example, 16p11.2, another name for a copy number variant that seems to be associated with potentially risk for a whole variety of different conditions. And we began to, and now these again are rare variations. They're not thought to be involved in most cases of schizophrenia, for example. But a number of studies began to report that similar overlap might be true for even the more common variants. And I'm not going to walk you through the details of this slide, but this was these were results that came out back in 2009 from the International Schizophrenia Consortium. And the bottom line is what they were able to do by studying the genome was essentially create a genetic risk score that involved the results of their genome-wide analysis of schizophrenia, and they asked whether, if you developed a genetic risk score from a genome-wide analysis of one data set in schizophrenia, would it be correlated with risk in another data set? And the answer was yes. But the more surprising thing was they said, well, if you if you calculated this genetic risk score in schizophrenia, would it tell you anything about the risk of bipolar disorder in a study of bipolar disorder? And the answer again was yes, it was significantly related. However, the absolute amount was still quite small, so it wasn't as though one could predict an individual's risk of either schizophrenia or bipolar disorder based on these results. But it did suggest for the first time pretty convincing evidence that variation across the genome that's associated with schizophrenia also seems to be associated with bipolar disorder to some degree. And that was consistent with what the twin studies I had shown you before were suggesting. There has been a remarkable collaboration in the scientific community in psychiatry and psychiatric genetics called the Psychiatric GWAS Consortium, or the Psychiatric Genomics Consortium. And this has been an effort to greatly enlarge the size of studies in psychiatric genetics to allow discovery of new genetic risk factors and new biology. And this is a somewhat outdated slide already, but there have been large-scale studies of major depressive disorder, bipolar disorder, schizophrenia, ADHD, autism, where researchers from around the world have come together to try to do more than anyone could do on their own and to really enhance the power of the studies that everyone has been doing. And this has been really quite successful. There have been a number of publications that have emerged from this consortium that have identified new genetic loci that are associated with schizophrenia, with bipolar disorder, and so on. And I wanted to talk about one analysis that the consortium has done which bears on this question of the relationships among these disorders, the boundaries. And that was part of the a subgroup of this consortium called the Cross Disorder Work Group. And that analysis was asking the question, if we actually thought about all of these disorders at the same time, would there be any risk factors that actually confer risk across these diagnostic boundaries that are not specific to, say, bipolar disorder or ADHD, but might actually, in fact, be related to more than one? And so what we were able to do was to ask two questions, really. What kinds of findings would we have if we did a combined analysis of all 5 of these disorders, which we think of as really quite different, but are there any shared genetic risk factors? And then also for risk variations that had previously been associated with, say, bipolar disorder, what pattern of effects might play out if you looked at those variants in other disorders? And this was a paper that was published about 2 months ago now, in which data for 61,000 or more cases and controls across these 5 disorders was put together. And this picture, this sort of picture that you're seeing in the graph here below the table, is a summary of results, and I'll just tell you what this is. It's not important to obviously pay attention to the details, but just so you know what you're looking at here. Across the bottom are each of the chromosomes in order, 1, 2, 3, 4, 5, 6, all the way up to 22. And each of these little dots, the gray, black, and red dots, represents a genetic marker that was tested across all those different chromosomes. And then the y-axis, this part that goes from 1 to 14, represents the strength of the statistical association with these 5 disorders in this kind of combined analysis. And the red line that's cutting through the middle of that graph is where we set our threshold for declaring something really statistically significantly associated. So what you see is that there were several regions that appeared to have markers that were associated across these 5 disorders to some degree. And that was interesting and in some ways surprising, although as you had seen from the previous family studies and twin studies, we did have evidence that there would be some genetic factors involved. The copy number variant studies suggested that there would be overlap of some genetic risk factors. But here's another example where you've got specific genetic regions and variants that seem to have effects across even these 5 disorders. And in fact, these are a couple of tables from the paper which, again, are not worth looking at in great detail, but just to orient you to it, the top table is answering that question of what would we see if we put all these 5 disorders together. And all the way on the left side are the names of markers, RS253, 5629, not particularly meaningful, I'm sure. But at the right side, what we have is the best fitting model, the best interpretation of the evidence based on the data we had for what this disorder was associated, or what this marker was associated with, or this region that the marker is in. And for the 3 of those 4 that were significant across the genome, they were associated with all 5 of the disorders that we looked at. They appeared to be. One of them seemed to be more specific to bipolar disorder and schizophrenia. And then the bottom table has that second question of what if we looked at variants that had already been associated with, say, bipolar disorder or schizophrenia, would they be associated with other of these 5 conditions as well? And so if you look, what you will see is for the bipolar disorder risk variants, which are the ones, the 4 at the top there, 3 of the 4 seem to be most likely to be specific to bipolar disorder. 1 of them seemed to have effects on bipolar disorder, depression, and schizophrenia, major depressive disorder and schizophrenia. For the schizophrenia variants in the bottom of the table, several of them seem to have effects that were beyond schizophrenia itself. And we also did another kind of analysis which was similar to the one I showed you between bipolar disorder and schizophrenia, involving these genetic risk scores. And the bottom line of this, without going too much into the details, was that there did seem to be, again, when you put together many of the variants at a time, a correlation between genetic risk for schizophrenia and bipolar disorder, between depression and bipolar disorder, to some degree between autism and schizophrenia, much lesser degree. But the bottom line of all of this is that the boundaries were not so sharp as we might imagine. And now we're beginning to see that at a actual genomic level. One of the more interesting and I think hopeful kinds of analyses one can do that was done as well in this large consortium study was to actually look at what we call pathway analysis, was to look at groups of genes and say, are there any biological pathways or biological functions that seem to carry most of this association with a disorder? And what we found was that, in particular, genes that were involved in how calcium channels operate in the brain, how they handle calcium, how neurons communicate using calcium channels, these are the kinds of genes that we're talking about, seemed to be associated with risk of all 5 disorders. And that kind of biological information is particularly interesting, I think, because it points to the possibility that we might learn something new about targets for treatment, about understanding the biology, how it works, what are the biological pathways involved, but also can we develop new therapies, medication therapies or other therapies that target some of these new biological clues and might be more specific and effective. And in fact, as people probably know, most of the psychiatric medications that are available today are really traceable back to biological insights that are more than 40 years old. Lithium, of course, has been around for quite some time. The antidepressants are based on a sort of set of biological insights that are still quite older. Same thing for the antipsychotics and anticonvulsants. And so the hope here is that we might actually learn something more fundamental that might allow us to develop really a new set of interventions that might be more specific or more tailored, perhaps, to what's actually going on in terms of the disorder. So just to bring us back to the questions we asked at the beginning, I think we can say that we do have pretty strong evidence that genes contribute to bipolar disorder. We also know that the contribution of any individual gene is not at all like what we think of in classical genetics. That is, this is complex. There are multiple genes that are involved that are expressed differently in different environments, and so it is really a complicated picture. But we have been able to find genetic variations, I should say genetic variations rather than genes, to be more specific, that appear to influence the risk for the disorder. And I think, interestingly, it's beginning to tell us something about the boundaries of mood disorders and about the relationship of psychiatric disorders. As you know, the current system, based on the DSM, really classifies disorders based on constellations of symptoms that have been grouped into syndromes, rather than on something about their underlying causes or the risk factors that go into them, or how they work at a more fundamental level. And that's one thing that we may see as a dividend of this research as well, a deeper understanding of how to diagnose, how to think about, classify, and ultimately treat bipolar disorder and other conditions. One question that comes up is, will DNA trump DSM? That is, will we have some kind of genetic test that really tells you whether you have one condition or another? I think the answer to that is no, at least in the sense of, because again these things are so complex, the genetics tells us something about risk. It tells us something about biology. There is nothing like a diagnostic test at this point, at least. But also, the way we think about disorders is not purely based on one set of causes. And there are many examples in medicine, for example, ulcerative colitis and Crohn's disease, which actually share quite a substantial proportion of their genetic risk factors in common, but we recognize those as different disorders or diseases. They also share things with rheumatoid arthritis. That doesn't mean that those are the same disease. So when we get to the concept of how we begin to define these things, it becomes more complicated, but I think fascinating. And I think this move towards getting deeper at a more fundamental level about how the brain and the mind work is really the way to go to help us understand how we should be thinking about the boundaries between disorders, when a disorder can be said to occur versus just variation that we might expect to see in the normal range, and so on. And in fact, I'll just mention that for those who are interested in those kinds of questions, I recently published a book on this topic, and I'd invite you to take a look at that as well. It's called The Other Side of Normal. But I'm going to wrap up and thank a few colleagues, especially in this cross-disorder genetics group from around the world. This is really a large consortium of 400 or more investigators, so this is a small fraction of those who really contributed. But it is really a pleasure to have the opportunity to talk to you today, and I really look forward to trying to answer any questions that you may have. So thank you again.
Thank you so much, Dr. Smoller. We're very appreciative. Our first question is, can you comment on epigenetic factors related to mood disorders and embryo pre-implantation genetic diagnosis for mood disorders becoming available in the future?
Wow, interesting question. So for those who may not have been following the story of epigenetics, what that refers to is the fact that in addition to our genetic code, our DNA code, there is a parallel code called the epigenome. And that has to do with things that influence the expression of our genes that are not coded in the DNA itself. So, for example, chemical markings that can change the chemistry of the chromosomes in ways that alter how genes are turned on and off and where they're turned on and off. And there's been a lot of interest now in this area because, of course, genes, the action of genes is through their expression. They have to be turned on or off to matter. And some of these controls, these sort of dimmer switches that can get attached to the DNA, may have big effects on risk of illness, for example. And this is, again, an area that is really very much in the early stages of research. One of the reasons that people are fascinated by it is that environmental exposures and experiences may be part of what can change the chemistry of our chromosomes. And there has been now the beginning of work suggesting that some of these epigenetic marks are correlated with risk of illness. But again, this is at an early stage. And one of the things that makes this area tricky is that the epigenetics of one tissue may not be the same as another tissue. So mostly people have not studied the brain for epigenetics because we can't actually study the brains of people who are living, of course. And so people have looked at epigenetic changes in the blood or the saliva and found some correlation with risk for mood disorders, for depression, for schizophrenia. But it is still a little bit hard to know how that relates to what's going on in the brain. And so there are a lot of technical issues that need to be sorted out. Whether that will ever be predictive enough for some kind of diagnostic value, I think is hard to say. I would tend to doubt it because just like the genome itself, these things are complicated enough that they certainly wouldn't be deterministic of risk for most people. Although it is possible that putting together genetic and epigenetic information, we might know something about whether somebody's at higher or lower risk.
Thank you. Given an influence of the environment, physical and social, on genetic expression, what about the reverse genetic expression affecting environment? How dynamic might such an interaction be?
Yeah, well, that gets to two questions. One of which is related to what I was just talking about, which is that the environment really has, as not a surprise, has an effect on how our genes are expressed. And there's been some fascinating work done largely in animals at this point, but suggesting that, for example, early adversity, trauma, and other related things can affect how active genes in the stress response system may be, and that those changes may be long-lasting. So there is evidence, we certainly know from certain toxic exposures that things like smoking and so on can affect the epigenetics of our genome and how active certain genes are or inactive. There's also another element to this which is fascinating, which is that the experiences and environments that we are exposed to are in part correlated with our genetics. And I'll give you an example of that. It turns out that the heritability, kind of the genetic differences contributing to, say, trauma exposure is about 40%. That is, there's a genetic component to whether somebody might experience certain kinds of experiences. And you can imagine that correlation between genetics and environment as occurring for a number of reasons, one of which might be, gene effects on personality, on risk-taking, or on other things that might make it more or less likely that somebody would be exposed to certain environments. So I think from both sides we are in fact seeing a kind of dynamic interplay between genes and environment. And we can't really ever separate those things because it's not really meaningful to say that, something is, this part is genetic, this part is environmental. It is always an interplay.
Thank you.
Is there another question?
Yes. Our next question is, have studies looked beyond first-degree relatives to determine if, say, a grandparent or uncle might contribute any genetic risk factor?
There have been studies of larger families. They are much harder to do, and so there have been much fewer. To the extent that they've been done in bipolar disorder, the evidence is that there is quite a drop-off after the first-degree relative. Instead of say a 10-fold increased risk, let's say for a first-degree relative, it might be down to more like a 2 to 3-fold increased risk for a second-degree relative. One thing I should say about that is those studies are based on a set of families that have been enrolled in the research, and as we all know, all of our families are different. We have different patterns of other comorbid disorders in the family and so on. So it's hard to get a lot of precision there. But it does seem like there is a drop-off quite a bit when we get to the second-degree relative. But it's still, significant compared to just the general population risk. And the thing that's also important to think about when you think about risk is, I mentioned that for, say, bipolar disorder, first-degree relative might have, say, an 8 to 10-fold increased risk of the disorder compared to the general population. But you also have to remember how frequent the condition is when you think about that. So that's a relative risk. But if the condition affects, say, 1% of people, that means that one might be going from a risk of 1% in the general population to, say, 9 or 10%. That is, there would be a 90% probability that one wouldn't be affected. The other thing about risk, familial risk for bipolar disorder, is that there is also actually an increased risk for just unipolar or major depressive disorder. So in fact, because depression is more common, it's more likely that somebody who's the first degree relative of somebody with bipolar disorder would suffer from depression than bipolar disorder itself. So once again, I think that also underscores the fact that these things don't exactly follow very specific categories.
Our next question is, how soon do you believe there will be enough genome data available for a person to have a blood test to determine whether an embryo has the genetic code for bipolar disorder?
Yeah, I think, I don't, personally, I don't really foresee that, at least not from where we stand today, for a few reasons. One is, as I mentioned, the genetics are very complex. The individual risk factors that we know about explain a small portion of risk, and therefore would be, not terribly predictive at this point. You could imagine a scenario, I guess, where as we have, large whole-scale genome sequencing and there's a lot more data down the road, that you might be at least able to say probabilistically that somebody would be at higher versus lower risk. And that might be true for things like heart disease and asthma and diabetes and so on. Whether people would want to have that kind of information and use it as a test, knowing that it was not, it was far from definitive, is one that I think is going to require, if we get to that point, a lot of discussion, a lot of thoughtfulness, and a lot of understanding of both the limitations and meaning of that kind of information. You also have to consider, what would one do with that information? Would it be, something that one would act on in some way, or, preventive treatments are not really very widely established or available. So there are all kinds of complicated issues attached to that. I think just from the scientific standpoint, we're not close to that at this point, just based on what we know.
Thank you. We have time for one last question. So I apologize that we couldn't get to everyone, but our last question is, how do you know if something like depression is caused due to genetics, or because of the factors, or because factors brought out the genetics? Is there a way to tell?
There isn't. There isn't at this point. And in fact, going back to something I was saying before, we think in general that the most common scenario is that there is some kind of combination of risk factors, that one might have some genetic vulnerability, and that in the face of certain environmental experiences, that vulnerability might translate into actual disorder. That's not to say that's always the case, and there's certainly situations where one might, the strength of the adversity that one is facing, or for whatever other reasons, regardless of genetic vulnerability, the environmental risk factors seem to be the trigger. Or conversely, where somebody might have such a high degree of genetic vulnerability that it's really hard to identify some specific precipitant. But the bottom line is that at this point we wouldn't be able to tease those apart. There's a lot of research that's going towards trying to understand that better, gene-environment interaction, as we call it. But we still know quite little about that.
Thank you so much, and thank you everyone for joining us today. Please remember that All our webinars and lectures are free, so if you'd like to make a donation, you can on our website at www.internationalbipolarfoundation.org. And thank you again, Dr. Smoller.
Thank you so much.
Bye-bye.
Bye.